Today's lecture deals specifically with cellular respiration. In our last lecture, we talked quite a bit about ATP and how ATP functions as an activated carrier. Its job is to take energy from reactions that release energy and transfer that energy to those reactions that require an input of energy. Which leads us then to the process known as cellular respiration. Cellular respiration is a catabolic energy-yielding pathway in which oxygen and other organic fuels are consumed and ATP is produced. So again, this is a catabolic pathway, meaning a pathway where large molecules are broken down to produce smaller, simpler molecules, but more importantly, there is a release of energy. Cellular respiration is an aerobic process, which means it requires oxygen in order to proceed. We can look at a variety of summary equations for this mechanism overall. And the most simple equation is shown right underneath the title, Summary Equations. This is where, again, organic compounds or fuel sources plus oxygen in a series of steps results in the production of carbon dioxide, water, and most importantly, the release of energy. We can also look at the equation just below that one with a little more specificity. Typically, when we talk about cellular respiration, we focus on glucose as the food or fuel source that is going to be utilized in harnessing or extracting energy. So in this equation, you can see that glucose plus oxygen, again in a series of steps, will result in the production of carbon dioxide, water, and most importantly, energy in the form of ATP. Now, before our transition into these alternative learning formats, we did have at least one lecture where we discussed ATP, and I reviewed that for you in the lecture that is already posted on Blackboard. In addition, we did begin the discussion of cellular respiration. If you recall, we looked at this particular slide, and the concept here is really just to provide you with an analogy. In order to make your car move, you have to provide it with gasoline or fuel. So in this example, our fuel source is gasoline. It turns out that it also does require oxygen for the process of combustion. The process of combustion results in the release of energy in the form of heat, but also the conversion of some of that energy into kinetic energy, which allows for the movement of the wheels to take place. You can also see that carbon dioxide and water are what are considered waste products in this reaction. So similarly, inside of a cell, we have food or fuel sources that are utilized in the presence of oxygen. The mechanism here of energy conversion is the process we're beginning to discuss, known as cellular respiration. There is some energy that is lost in the form of heat. However, a lot of the energy is converted into this form known as ATP, and that ATP can then be utilized in the cell for functions that require the input of energy. Again, you also see that the waste products in this reaction are carbon dioxide and water, similar to what we just talked about with the car example in the panel above. Okay, so let's look at cellular respiration in a little more detail. What happens overall in the series of reactions is the mechanism allows for the oxidation of glucose, the energy source or fuel source. The energy is taken out of the molecule known as glucose and is converted into a form known as ATP that can be then utilized by the cell. So again, what you see in the center of this slide is a summary equation where glucose plus oxygen in a series of steps will lead to the production of carbon dioxide and water. And what is not on this figure is the production of ATP, which again is probably the most important point behind cellular respiration. It is a series of redox reactions, and what is happening in these redox reactions is glucose is undergoing oxidation. It is losing those high energy electrons, and oxygen is undergoing a reduction. Oxygen is gaining eventually those high energy electrons. Now, again, previously we talked about free energy diagrams, and we utilized this example of free energy diagrams associated with a hydroelectric plant. We discussed the fact that having a simple system where one reservoir is at a higher level than the next reservoir, in between those two reservoirs we have a turbine that, as the water leaves reservoir 1, enters reservoir 2, that turns the turbine and allows for the conversion of that kinetic energy to light the light bulb. However, in a closed system, eventually the water will reach equilibrium and no more electricity can be generated. We also discussed that a way to make this system better is to make it an open system. An open system allows for the continual flow of water in and the removal of the water from reservoir 2. This allows for the water to continually flow, turn the turbine, generate electricity. Lastly, in the bottom panel, we discussed that we could make this system even better. The way we can make this system even better is to have a series of reservoirs or tanks, each one at a slightly lower level than the one before it. Also utilize an open system where water can continually flow in reservoir 1 and exit reservoir 4. In this case, as long as there is a continual supply of water, all of the turbines will turn because the water moves downhill towards tank 4. By making it a multi-step system, we are now generating more electricity rather than in just one step. This is exactly what is happening during cellular respiration. It is a multi-step process, and by having it be a multi-step process, the maximum amount of energy can be extracted. Okay, so now let's look at an overview of the entire process. Cellular respiration is divided into 3 metabolic stages. The first one is known as glycolysis, and that's what you see on the far left in this figure. Glycolysis is a mechanism that occurs within the cytosol or cytoplasm of the cell, and this is where glucose undergoes a series of steps to be broken down into a molecule known as pyruvate. In this process, there is some ATP that is generated. However, there are a number of energy conversion steps that are also occurring in this mechanism, where high energy electrons are being transferred from to NAD to produce NADH. That NADH now has a great deal of energy associated with it because it has picked up those electrons. We'll talk about what happens to NADH in just a little bit. The process or stage that you see in the center is known as the Krebs cycle. The Krebs cycle occurs within the mitochondrion. Remember, the mitochondrion was considered the powerhouse of the cell, and this is where the site of ATP production occurs. In the Krebs cycle, there is ATP production. However, there is also that mechanism of high energy electron transfer to NAD to produce NADH. Lastly, also occurring in the mitochondrion, on the far right-hand side of the slide, you see what is labeled as the electron transport chain. This is a mechanism where the NADH that was generated in glycolysis and the NADH generated in the Krebs cycle is all transferred to the electron transport chain in the eventual production of ATP. ATP can be produced by two different mechanisms in the entire cellular respiration pathway. One way to synthesize or produce ATP is known as substrate level phosphorylation. Now, in substrate level phosphorylation, you have an enzyme that is going to bind two substrates in the transfer of a phosphate group from substrate 1 to substrate 2, which happens to be ADP. So remember that cyclic process we talked about in the last lecture where ATP has a great deal of energy, but once the phosphate group is removed, it becomes ADP. ADP can be converted back into ATP by adding a phosphate group back on. Substrate level phosphorylation is the mechanism that does just that. It takes a phosphate group from one substrate, adds it to ADP to produce ATP. This is a direct synthesis of ATP. The second way that ATP is synthesized during cellular respiration occurs specifically within the electron transport chain and is known as oxidative phosphorylation. It is a mechanism that allows for the production of ATP using energy from redox reactions. Those NADH molecules that we talked about that are now holding on to those high energy electrons are utilized in oxidative phosphorylation to lead to the eventual production of ATP, and we will look at that in more detail in just a minute. Oxidative phosphorylation is not a direct synthesis like we just discussed with substrate level phosphorylation. It's an entirely different type of mechanism. The first stage, or metabolic stage, of cellular respiration is glycolysis, like we discussed in that summary slide. Glycolysis is a mechanism that itself is multi-step and is divided into two phases. There is what's known as the energy investment phase, meaning that there does need to be a slight input of energy to allow the reactions to proceed. The second half of glycolysis is known as the energy payoff phase, and this is because energy is produced in the form of ATP as well as NADH in the energy payoff phase. If you look at the bottom of the slide, you can see an overall summary of what's happening in glycolysis. Glucose enters the pathway, 2 pyruvate and 2 water molecules are produced. In addition, there is a requirement of 2 ADPs and 2 phosphates that will lead to the eventual production of 2 ATP. And then on the last line, you can see 2 NAD are required, which will result in the synthesis of 2 NADH. Now, if you look at the overall equation, you can see that in the energy payoff phase, 4 ATP are produced for every 1 glucose that enters the pathway. However, in the energy investment phase, 2 ATPs are utilized. So if you make 4 and you use 2, that leaves a net of 2 ATP for every every glucose molecule that enters glycolysis. Now let's look at the energy investment phase in a little more detail. I will never ask you to tell me every single step of glycolysis. The summary is what's important. However, I always like to review the steps and stages just so that you get a good understanding of what is happening. Again, you can see that the energy investment phase is a multi-step process. Glucose enters into the pathway, and the very first enzyme that is encountered is a molecule, an enzyme known as hexokinase. Specifically, kinases are families of enzymes that transfer phosphate groups from one substrate to another. This is the very first example of where ATP is required. The phosphate group from ATP, the terminal phosphate group, is removed in this step and added to glucose. This step is also really important because it traps glucose inside of the cell once the very first step takes place. Again, the arrow now indicates another kinase. This is phosphofructokinase. However, it again is a kinase. A kinase takes the phosphate group from ATP and transfers it to another substrate. Next comes the energy payoff phase. And here too, again, I'm not going to ask you for every single step. I just like to highlight this to give you some idea of the complexity that is taking place during this process. In particular, you can see that there are enzymes that are kinases. Kinases, again, transfer the phosphate group from ATP to a substrate. Hence, ATP is required at that particular stage overall. You also see pyruvate kinase, again, one of those steps where a phosphate group is utilized or taken from ATP overall. But lastly, what I want to highlight is an enzyme that is known as a dehydrogenase. A dehydrogenase, or dehydrogenases, are families of enzymes. These family of enzymes are involved in electron transfer. We won't get into the detail of how that electron transfer takes place. What's important is that dehydrogenases play a role in transferring electrons from the substrate to NAD to produce NADH. Remember again, NADH now has a great deal of energy associated with it because it has accepted those electrons. The next metabolic stage is known as the Krebs cycle, and the Krebs cycle takes place within the mitochondrial matrix, that is the innermost component of the mitochondrion. However, pyruvate, which is one of the end products of glycolysis, needs to pass through the mitochondrial membrane system and enter into the matrix. So this step that you see on the slide on the left-hand panel is actually that step of getting pyruvate into the mitochondrion. It's not actually part of the Krebs cycle. However, in this case, what you can see from that figure is that pyruvate does in fact enter into the mitochondrial matrix, and there are a series of steps that takes place. Step 1 is the removal of a carbon in the form of carbon dioxide. Step 2 is the production of NADH, again, specifically electron transfer that occurs by an enzyme known as pyruvate dehydrogenase. And then step 3 is the addition of a molecule known as coenzyme A to produce acetyl-CoA. Acetyl-CoA now can enter into the Krebs cycle. So on the right-hand side, you can see a summary of the Krebs cycle. On the top portion is what we just discussed, that is getting pyruvate into the mitochondrion with that series of steps. And then the circle or cyclic portion is the Krebs cycle. With the Krebs cycle, in the summary, what you can see is that there is the release of carbon in the form of carbon dioxide. There is the production of NADH. Again, electron transfer to NAD will produce NADH. There is also a small amount of ATP produced directly in the Krebs cycle. And then lastly, you can see that there's another high energy molecule that is produced. FAD is a molecule very similar to NAD. It is an activated carrier. It too can pick up electrons. It becomes FADH2, and FADH2 now has those electrons associated with it and has a great deal of energy associated with it. So let's look at the Krebs cycle in a little more detail. Just like I mentioned for glycolysis, you do not need to know every single step, but I like to always go over and highlight a few things just so that you get a better understanding of the complexity of the process. Up at the top of the cycle, about 12 o'clock position, you see acetyl-CoA entering into the cycle. There are a series of steps that take place during the Krebs cycle, but what I want to highlight again for you are the enzymes that are known as dehydrogenases. Dehydrogenases, again, allow for the electron transfer to NAD to produce high energy NADH. There you see it again in step 4 and step 6 as well. Let's move on to the last metabolic stage, which is known as the electron transport chain. There's a few things that I want to highlight with this particular figure before we jump into the mechanism of the electron transport chain. The first thing I want to highlight for you in the right-hand corner is the figure of the mitochondrion. Now, if you remember when we discussed mitochondria in general, we talked about the fact that it is made up of a double membrane system. There is an exterior or outer membrane, and there is also an inner membrane. The inner membrane is folded on itself back and forth, generating very high surface area, specifically for those enzymes that are part of the electron transport chain. It's essentially giving a platform for all of those enzymes to dock or bind to. So in this figure, the same mitochondrial figure, you have a darker orange color and you have the lighter orange color. The lighter orange color represents what's known as the mitochondrial matrix, and the orange color refers to or highlights what's known as the intermembrane space. It is the space between the outer and the inner membranes in a given mitochondrion. If you now look at the larger portion of this figure, you can still see that orange and lighter yellowish color highlighted in the figure. Across the center or the middle of the figure is the inner mitochondrial membrane. And what you see are some of the components of the electron transport chain that are embedded in that inner mitochondrial membrane. And again, just to give you some visualization of space, on the top half of that figure is the inner membrane space, that orangish color, and then below is the mitochondrial matrix in the lighter yellowish color. So all of that NADH and FADH2 that was synthesized in glycolysis and the Krebs cycle is shuttled to the electron transport chain. Now remember, those molecules, NADH and FADH2, have a great deal of energy associated with them. What happens during the electron transport chain is those high energy molecules, NADH and FADH2, transfer their electrons to components of the electron transport chain. Now, because of that transfer of electrons, that has a great deal of energy associated with it. what happens with that transfer occurs simultaneously, and that is the passing of a hydrogen ion or a proton from the matrix into the inner membrane space. And it is this process that is referred to as chemiosmosis. Chemiosmosis is the ability of certain membranes to use chemical energy to pump hydrogen ions and harness the energy to do work. In this case, the work is to synthesize ATP. So I've highlighted for you where NADH enters the pathway, and on the next slide I will show you where FADH2 enters the pathway. The important thing to note here is that there is a continual transfer of electrons through all of the components of the electron transport chain, which then allows for hydrogen ions to be pumped across the membrane, generating a concentration gradient of hydrogen ions in the inner membrane space. It is this high concentration of hydrogen ions in the inner membrane space that then causes or allows for the production of ATP. So let's look at it again with that same figure as well as an additional figure, just giving you a different frame of reference. We talked about how NADH enters the pathway at the beginning of the electron transport chain. FADH2 enters at a slightly different position, and so not as many hydrogen ions get pumped across the membrane for every FADH2 that donates or transfers its electrons. So what we can do is a rough estimate of how much ATP can be produced overall through the electron transport chain. And the way we can do that is just by simply looking at electrons that get transferred from NADH or from FADH2. So for every NADH that transfers an electron to the electron transport chain, 3 hydrogen ions get pumped across the membrane. And you can see that 3 starting with the first position, the middle, and then the last position. For FADH2, because it enters at a different point in the pathway, that very first pump is bypassed, so only 2 hydrogen ions get pumped across the membrane for every FADH2 that donates electrons.